Hello biologists, this is Enzymes Digestion Part 2. Let's recap the results of the milk digestion. This is a glucose strip that shows no glucose detection. It's kind of bluish. This is a glucose strip that shows glucose detection. It's kind of brownish. Here they are together. This brown one here, this one, shows that glucose is present. This little bit of reactant paper here is impregnated with an enzyme that detects glucose, that actually reacts with glucose, and the product of that reaction is detected by another chemical that turns a color. So that's how glucose is detected in milk. Remember, just plain old milk didn't have any glucose in it. There was no glucose. We got the blue strip, and that showed that lactose was not digested into glucose. When we got down to milk here that had lactase added, remember lactase is an enzyme that breaks down lactose. Lactose, remember, is a disaccharide. It has two sugar rings here, and when it gets cut by lactase, the enzyme breaks it into two monosaccharides, two separate sugars. One of them is glucose that's picked up by these detector strips. So when this happens, when this lactose, that's two of these stuck together, gets broken into two monosaccharides, we detect the brown on the strip. And so when we add lactase, we get glucose because we cut the big sugar into two little ones, one of which is glucose. The detector can't detect when the two sugars are stuck together. This doesn't react with the detector strips. When lactase chops it in two, then it reacts and we get the brown strips. So let's talk about some of the lab questions here. Did the digestion of the milk match your prediction? Well, go back to the table we filled out in the very beginning where you predicted what was going to happen and see if the results support your hypothesis. What happened to the lactose here? What happened to the big disaccharide? The two sugar, di means the same thing as bi, and you know you ride a bicycle with two wheels. So what happens to this big sugar here? What does the lactase do to the lactose? This is lactose, the big sugar here. Which test tubes tested positive for glucose? And what does this tell you about lactase and lactose? Well, we can go back to table 4 here on your lab sheet to answer question 2 on your lab sheet and look at here. We know that there's no glucose here, there's just lactose in 1 and 2, and in tubes 3 and 4 we added lactase, and so we got some glucose digestion. And so we know that lactase, the enzyme, acts on lactose, the sugar. So question 3 on your lab sheet says, describe the appearance of the test tube with the most milk digestion. What was responsible for the digestion? Well, let's go back up to the lab results here. I took a screenshot, and this is what shows the lab. It doesn't really look any different. The test tubes with lactase don't look any different, except when you use these detector strips. So that's what you have to pay attention to when you're looking for the digestion or the breakup of lactose. What was responsible for the digestion is lactase. Enzymes don't change the appearance of what they digest, but lactase does digest lactose into the monosaccharide glucose. Okay, question 4. Let's learn a little bit about the stomach before we get on to question 4, which is about the second digestive enzyme in our experiment today. The stomach kneads and grinds food and also adds an enzyme called pepsin. Pepsin is secreted by the stomach. It's in the mucosal cells of the lining of your stomach right here. And it breaks down proteins. Pepsin also acts as a molecular scissors to chop up the big proteins, the long amino acid chains that we've been talking about. These long amino acid chains, even longer than this, several hundred amino acid chains. If you had maybe a steak for dinner, you have some big proteins in that meat, lots of amino acids all stuck together. Your body can't absorb the big polypeptides, the big proteins, so pepsin acts to cut those amino acid chains into smaller polypeptide chains so your body can absorb something like this in your small intestine. So pepsin is a molecular scissors. It catalyzes the breakdown of proteins into shorter amino acid fragments. It turns the big long fragments into shorter ones. And this happens in your stomach. The stomach happens to be a pretty acidic place, and we talked about that when we talked about lactose. Lactose works in a semi-acidic environment. It works in your small intestine, but pepsin, which works in your stomach, has to work in a pretty darn acidic place. Now, you know your stomach, we talked about this before in part 1, you know your stomach's pretty darn acidic. If you've ever puked and had it go up your nose or in the back of your throat, it's pretty nasty stuff. In fact, it has a similar pH to Limaway, which is this cleaning product that you use to get rid of the calcium deposits in your shower. So pepsin has to work in a pretty low pH. And in fact, if you buy pepsin, I don't recommend you do this, but some health food stores sell pepsin, the enzyme, to help people with digestion problems. It comes with HCl added to it, the same stuff we're going to add in the experiment, the same stuff that your stomach adds. Hydrochloric acid is the same as HCl here, because the enzyme pepsin works best in an acidic environment. Pepsin is secreted in the stomach. Pepsin has to work down here on the pH scale. Remember when we talked about proteins, which are basically a string of amino acids all put together? And some of those amino acids have side chains. And those side chains bond with each other. And that bonding between the side chains in a molecule, or in a particular protein molecule, gives the molecule its specific shape. And molecular shape is really, really important for enzymes. If you put them in the wrong pH or at the wrong temperature, they lose their shape and they don't work anymore. They don't catalyze the reaction because their shape, this shape is going to fit really well around proteins to break them up, to act as a molecular scissors. But if we put this in the wrong pH, we put this in, say, a basic environment, we put this in drain cleaner or something like baking soda, which is basic, it's probably not going to work too well because some of these bonds might come undone and our nice shaped enzyme might turn back into a long chain and no longer catalyze reactions. So enzymes are kind of fussy molecules. They're not alive, they're just molecules, but they work best under particular conditions of pH and temperature. So here's the lab. We're going to try to digest some eggs with the enzyme pepsin, and we're going to look at different conditions under which those eggs digest. before we look at the experimental results, we're going to make some predictions based on what's in each test tube. In one of the test tubes, there's just water and eggs. There's pepsin in one of the test tubes. There's hydrochloric acid. There's sodium bicarbonate. In one, sodium bicarbonate is just what's in baking soda. That's a base. It's the opposite of an acid. There's pepsin and sodium bicarbonate, so we're going to see if pepsin works in a basic environment. And then there's hydrochloric acid, which is basically what's in lime-away. Well, actually that's sulfuric acid, but it's a similar pH to lime-away or really concentrated vinegar. So knowing what you know about enzymes and how they work, and specifically how pepsin works and where it's supposed to work in the stomach, which is a pretty acidic environment, more like vinegar than baking soda, make some predictions on your lab sheet. Fill in this table based on if you think the egg white will be digested. Just so you have a close-up view of what eggs in a test tube look like, this is eggs in a test tube with just water and no enzyme, and they've actually been sitting around for a couple of days now. So it's just plain egg white. I chopped up, put in a test tube, put some water in there. Not much is going on. I filled out the pH table for you using good old regular pH paper, much like in the slide here. I took the pH of the test tubes in the virtual lab and the test tubes in real life as well. And the test tubes with just water in them have a neutral pH, kind of in the middle. That's where water should be. Test tubes with hydrochloric acid in them end up around 3 on the pH scale. That's down in the acid range here. Test tubes with baking soda here end up in the basic end of the scale. Not super basic, but right around here. And then when we add acid and the enzyme, it ends up around 3, more like your stomach. So there's a close-up view of pH paper if you need to see that. That's kind of how you read pH paper. If it's this color, it's pH 12. If it's this color, it's pH 1. So if you want to know the results, this is what happened at the end of the experiment. I went into the virtual lab, I set it all up, I let it sit for 48 virtual hours, and then went back to the lab and checked it out. And you can see the egg level in all of the test tubes except the one with pepsin and acid is exactly the same. Except this one, the eggs are down here instead of up here when we add the acid and the enzyme. Remember, pepsin works in the stomach, which is conditions very similar to having hydrochloric acid in the test tube. So here are the egg digestion results. This table is table 5, and write down a description of what happened to the eggs in this table. And I'll go back to this slide so you can check it out, pause the video, and record your observations. I have some test tubes here to answer your lab question 4. This is actually real life egg in meat tenderizer. I couldn't get a hold of any pepsin, but there's a enzyme called bromelain you isolate from pineapple that's in meat tenderizer. And I let these test tubes sit for a whole day, and I simulated conditions in the stomach, not only with the vinegar, but I also kept these closer to body temperature at about 35 degrees Celsius. So you can see this is egg with vinegar and meat tenderizer. This is meat tenderizer and water here, and this is with baking soda. And so the eggs got much more crumbly and kind of squishy at the bottom of the test tube when they were in an acidic environment with the enzyme. They were much more digested than when they were over here with baking soda. And just for comparison, this is just egg and vinegar, no enzyme. And here's the